So I have talked to you about the homologous recombination in prokaryotes. Uh, now I will try to discuss about the eukaryotic process of homologous recombination. Well, in eukaryotes, for the pairing of homologous DNA and strand invasion is carried out by the RAD51 and DMC1 protein. The full form of DMC1 is DNA meiosis recombinase 1. RAD51 is your DNA repair protein, RAD51 homolog 1, you can say them. And they are involved in pairing of homologous DNA and strand invasion. For the introduction of double stranded break in eukaryote, it is done by SPO11 during meiosis process. You will see them. And by the HO protein for the mating type switching when you will see. For the processing of DNA breaks to generate single strand for strand invasion, it is done by here by the MRX protein, also called as RAD50, RAD58, RAD60 nuclease in eukaryotes, which was actually done by REC-BCD in case of prokaryotes. For the assembly of strand exchange protein, it is carried out by RAD52 and RAD59. Holiday junction recognition and branch migration is not known till now. And for resolution of the Holiday junction, perhaps Muse 81 and other proteins are involved in, in cleaving that joint and to resolve, resolute that Holiday junction. Okay. So let's get started now about the eukaryotic homologous recombination process. So I have discussed about the prokaryotic homologous recombination. So homologous recombination in eukaryotes. So just we have described that homologous recombination in bacteria is required to repair the double stranded breaks in the DNA, to restart collapsed replication forks, and to allow cells chromosomal DNA to recombine with DNA that enters via phage infection or conjugation. In case of prokaryotes like bacteria, they need homologous recombination to repair that double stranded breaks if occur in the DNA, to restart that replication process due to collapsed replication forks, and to allow the cell's chromosomal DNA to recombine with the DNA that enters through bacteriophage infection or conjugation. But in case of eukaryotes, homologous recombination is also required for DNA repair and restarting of collapsed replication forks in eukaryotic cells. For the eukaryotic cells also, we need homologous recombination to repair the double stranded breaks in the DNA and to restart the collapsed replication forks in the eukaryotic cells. This requirement is illustrated by the fact that cells defect in proteins that promote recombination are hypersensitive to DNA damaging agents, especially those that break DNA strands. Right? So, homologous recombination in eukaryotes are specifically required for what? They are required for DNA repair. They also required for DNA repair and for the collapsed replication fork in the eukaryotic cells, right? But homologous recombination also required for during meiosis process. It plays an important additional role in eukaryotic organisms, like it is critical for meiosis. Unlike your with your DNA repair process, it is also involved in collapsed replication forks. Okay, but in eukaryotes, the most important thing of recombination is during meiosis. During meiosis, homologous recombination is required for proper chromosome pairing and for maintaining the integrity of the genome. This recombination also reshuffles the genes between the parental chromosome and ensuring variation in the sets of gene that will be passed on to the next generation just after the meiosis process. So homologous recombination is also needed during the meiosis process so that the proper chromosome pairing can occur, and it can reshuffle the genes between the parental chromosomes so that they can be, when the cell division will occur, when meiosis will occur, it will be passed on to the next generation. Now we all know about the meiosis process, that during meiosis two rounds of nuclear division occur. The first division is the reduction of DNA content from the normal content of diploid cell to the content present in the gametes. So there it is a form of the reductional division that normally occurs during meiosis. Before division, the cell has to copy each of these chromosome, one each that was inherited from its two parents. So before the cell division, during the interphase, the cell has to copy, the cell will have two copies of each chromosome, the homologs. Okay. During the S phase, these chromosomes are replicated to give a total DNA content of 4n, and the products of replication, that is the sister chromatids, will stay together. So in preparation for the first nuclear division, these duplicated homologous chromosomes must pair with each other and they will align among themselves at the center of the cell. Just like we have seen during homologous recombination, alignment of the DNA should be occurring, right? It is the pairing of these homologs that requires homologous recombination, and these events are carefully timed out. Recombination must be complete before the first nuclear division to allow the homologs, that is the homologous chromosomes, to properly align, and then they separate. During this process, the sister chromatids will remain paired. And then in the second nuclear division, it is the sister chromatids that will separate. The products of this division are four gametes, each having copies of each chromosome, that is the 1n DNA content. So recombination is required. It is required so that it is required. Why we required this homologous DNA? Why we do homologous recombination during cell division? Okay, for able to maintain the integrity of the genome and to reshuffle the genes between the parental chromosomes, that ensures variation in the sets of gene that will be passed on to the next generation. Okay. Now it is saying that without recombination, chromosomes often fail to align properly for the first meiotic division, and as a result there will be high incidence of chromosome loss. So if there is no recombination, if there is no recombination, then chromosomes will not able to align properly for the first meiotic division, and as a result there will be a high incidence of chromosomal loss that leads to non disjunction. just i have said here just I have said here that what happens during your what happens during your meiosis process. Wait for a minute. Okay, so for the preparation of the first nuclear division, the duplicated homologous chromosome pair and align at the center of the cell, right? And it is due to the pairing of the homologs that require homologous recombination. Okay, These events are carefully timed out. Then recombination must complete before the first nuclear division, and recombination has to be completed just before the first nuclear division of meiosis, that allow the homologues to properly align and separate. Okay, and during this process the sister chromatids remain paired. Then in the second nuclear division the sister chromatids again separate. So if a homologous recombination does not occur, then it will leads to what? It will leads to non-disjunction, which leads to failure of the chromosomes to enter to the gametes without correct chromosome complement. Gametes will have either few or it may have too many of chromosomes which cannot develop properly once fertilized. Thus, a failure in homologous recombination is often reflected in poor fertility. So, if homologous recombination does not occur properly, then it leads to poor fertility that has been stated. Okay. Now let us see that how homologous recombination, specifically the homologous recombination that occurs during meiosis, occur as follows. Okay, it says that the developmental program needed for the cells to successfully complete meiosis involves turning on the expression of many genes that are not needed for during normal growth, and one of these are SPO11. Okay, so SPO11 is one of the genes, and this gene encodes a protein. SPO11 encodes a protein that will cause a double stranded breaks in the chromosomal DNA to initiate the homologous recombination process in meiosis. So, the initial protein that involves during meiotic recombination process, the first protein which is involved is SPO11. So SPO11 is the first protein which introduces what? Double stranded breaks in the chromosomal DNA to initiate the recombination process. Then SPO11 protein cuts the DNA at many chromosomal locations with very little sequence selectivity, but at specific timing during meiosis. So at specific timing during meiosis, I think in the diplotin stage maybe, that at that time SPO11 protein perfectly cuts the DNA within the chromosome. At many chromosome locations it cleave. Okay. There is no such sequence selectivity. Okay. SPO11 mediated DNA cleavage occur right around the time when replicated homologous chromosomes start to pair. So this SPO11 mediated DNA cleavage occurs when the after replication occurs in the S phase, when the homologous replicated homologous chromosome starts to pair and align with each other. At that time SPO11 starts to carry out their main process to cleave at many specific locations. Okay. SPO11 cut sites, although frequent, are not randomly distributed along the DNA. Rather, the cut sites are located most commonly in chromosomal regions that are not tightly packed with nucleosomes, such as promoter controlling gene transcription. So, SPO11 cut at which sites? Which are not tightly packed, where nucleosomes are not tightly packed with the DNA. At that sites, SPO11 cleave, like the promoter region. These are the gene acting these are the activating areas na euchromatin areas where the transcription process normally occurs. So at that site SPO11 normally cleave. Regions of DNA that experience high frequency of double stranded breaks also show a high frequency of recombination. So those area which is a more chance of having double stranded breaks, they are also your SPO11 cleave at that sites. So most commonly used SPO11 DNA cleavage sites like the Chi sites are the hotspots for the recombination process. So SPO11 cleave where it cleave randomly but mostly they cleave in those chromosomal regions that are not tightly packed with the nucleosomes. And it also cleave those regions of DNA that have a high chance of double stranded breaks. Okay. Now, how your spo11 cleave the DNA? I have said right that spo11 cleave the DNA at specific position. Okay. It has not such specific sites, but it normally introduce double stranded breaks um, in two ways. Not in two ways. It can introduce double stranded breaks by observing non tightly coiled nucleosomes at the site. That means, what can I say? Those regions which are not tightly packed with the nucleosomes, like promoter controlling gene transcription, that side they can cleave. And those regions of DNA which has high chance of double stranded breaks, these sites can also be cleaved by the spo11. No, now molecularly, if we say in molecular mechanism how spo11 cleave, let us see. There is a specific tyrosine kinase, there is a specific tyrosine side chain in the spo11 protein, and that specific tyrosine side chain will attack a phosphodiester backbone to cut the DNA. So within the SPO11 there is a tyrosine amino acid residue. Obviously all proteins are made up of amino acid. The side chain of this tyrosine will cleave the DNA backbone. Okay, it will cleave the phosphodiester backbone to cut the DNA and generate a covalent complex between the protein and the DNA strand. Now there are two subunits of SPO11 that cleave the DNA two nucleotides apart on the two DNA strands to make a staggered double stranded break. So it actually cleaves the DNA strand in such a way to create a staggered breaks. A sticky end, to create a sticky end, a staggered breaks means to create a protruding end like this. It cleaves in this way to produce this type of protruding ends, sticky ends. Okay, SPO11 shared this DNA cleavage mechanism with the DNA topoisomerase and site-specific recombinase. Now, SPO11 cleave in such a way, it looks like that SPO11 cleaves the DNA. So, the function, the way that SPO11 cleaves the DNA is similar with the cleavage mechanism that is done by DNA topoisomerase and site-specific recombinase, right? And in fact, SPO11 appears to be found that it is a distant cousin of these enzymes, means related, far away. So, so within the SPO11, there is a specific tyrosine kinase, not kinase, sorry, there is a specific tyrosine side chain which attacks the phosphodiester backbone in the DNA and generate sticky ends, staggered double-stranded break. This way, SPO11 works. Now, this SPO11, when this SPO11 cleave the DNA ends, now it cleaves the DNA ends to generate these sticky ends, which became the initial site for the other DNA processing enzymes for homologous recombination, like REC-A proteins, and initiation for the strand invasion occurs. Just like in case of prokaryotes, we see the REC-A like proteins. Here, the MRX protein maybe you will see it will allow them to come and bind and interact. Okay, so let us see what they are trying to say here. So this is how SPO11 cleave. This is your double strand DNA, and this is a side chain of SPO11. They have not seen the tyrosine residue, and they are cleaving. They are cleaving to generate sticky ends, protruding ends, but staggered cut, and in this way they bind, which allows the other proteins to observe, and like the Reke-like proteins, which are called MRX protein, to come and bind and carry out the other process of homologous here meiotic recombination process. So about the MRX protein, what they are saying, the DNA at the site of SPO11 catalyzed double-stranded break is processed to generate single-stranded regions necessary for the assembly of a Reke-like strand exchange proteins. So the Reke-like strand exchange proteins comes and bind. As we observe that in the REC-BCD pathway for bacteria, this processing generates long segments of single-stranded DNA that terminate in the 3' end. Okay. So here also your the DNA at the site of SPO11 catalyzed double-stranded break, it has to be processed and generate single-stranded regions which are necessary for strand invasion, for strand exchange, and that is done by Reke-like strand exchange proteins. Here, MRX enzyme complex carry out the function during this meiotic recombination process. MRX enzyme is responsible for this DNA processing event. It generates long segments of single-stranded DNA that terminate at 3' ends for the purpose of strand invasion. Now, this complex MRX enzyme complex is not homologous with the REC-BCD, but it is a multi-subunit nuclease enzyme. MRX protein is composed of 3 protein subunits known as MRE11, RAD50, and XRS2. The first letter gives its name, okay? The first letters of these 3 subunits gave the name of MRX. Now, processing of the DNA at the break site occurs exclusively on the DNA strand that terminates at the 5' end. That is the strand that is attached by the SPO11 protein. So at the 5' end, it is been covalently attached by the SPO11 protein. Now the strand which terminate at 3' end have a OH group and they are not degraded. And this DNA processing reaction is therefore called 5' to 3' resection. MRX dependent 5' to 3' resection generates long single stranded DNA tail with 3' end. This MRX complex is also thought to remove the DNA linked SPO11. So MRX complex also remove the SPO11 link with the DNA at the 5' end. Okay. So this is the picture. You can see replicated homologs where SPO11 have come and bind, and MRX get that site to bind. It generates that sticky end, and it also removes the SPO11. The resection done by the MRX protein. MRX protein is made up of 3 subunits called as MRE11, RAD50, and XRS2. Okay, in this way, the resection of the strands done by the MRX protein. Then finally, DMC1, which is called as DNA meiosis recombinase 1. It is just like Reke-like protein that specifically function in meiotic recombination. Now, eukaryotes encode 2 well-characterized homologs of bacterial Reke protein, RAD51 and DMC1. These 2 proteins are homologs, similar like in structure. Both proteins function in meiotic recombination process, whereas RAD51 is widely expressed in cells dividing mitotically and meiotically. DMC1 is expressed only in cells which enter meiosis. So RAD51 is involved in mitosis as well as in meiosis. It is expressed in cells, whereas DMC1 is expressed on those cells which enter the meiosis. Strand exchange during meiosis occurs between the particular type of homologous DNA partner. Recall that meiotic recombination occurs at a time when there are four complete double-stranded DNA molecules representing each chromosome. Okay, so here we can see that pairing between the homologous partners occurs in this time. Although the two homologues likely to contain small sequence difference and carry distinct alleles for the various gene, the majority of the DNA sequence among these four copies of the chromosome will be identical. Interestingly, DMC dependent recombination is preferentially between the non-sister homologous chromatids rather than between the sister chromatids. So between the sister chromatids, DMC1 will not recombine rather than it will try to recombine between the non-sister homologous chromatids. Although mechanistic basis of this relativity is not unknown, there is a clear biological rationale. Meiotic recombination promotes interhomolog connections to assist alignment of the chromosomes for division. So in this way, your whole nuclear meiotic recombination occurs. You can see here DMC1 and RAD51, they bind. Okay, they bind here like Reke filament, and they cause the strand invasion here, just like the Reke does in prokaryotes and carries out the recombination process.